Yep, or GEDIT for short. So yes, I'm Brian. I'm from Matteo Pellegrini's lab. I'm a second-year PhD student. And so this tool, the basic problem that it addresses is tissue sample heterogeneity. So unless you're employing some sort of special purification procedure, every tissue biopsy ever collected is a mixture of multiple cell types. This can be, it can be informative to figure out these cell types. For instance, in the case of cancer, we've increasingly found that the number and type of infiltrating immune cells can correlate strongly with outcome and also with response to treatment. So this includes like chemotherapy and also these newer immunotherapy treatments. Also in laboratory experiments, changing cell populations can be a confounder in terms of your analysis, because if you see an observed change in gene expression, that could be due to true changes in gene expression, or it could in fact be due to changes in the underlying cell types in the populations. So quantifying this can be very valuable, but physical means of doing so are currently pretty limited. One way of doing it is cell flow cytometry, like this machine on the right. But it's expensive, it requires some expertise, and it's also problematic on solid samples. It requires that you get a suspension of single cells, and on solid samples, in order to get a suspension of single cells, it can sometimes be difficult to do that without damaging them. Laser capture microdissection kind of falls in this category as well. You can get some pretty pure samples, but it's very expensive and slow and requires some real expertise. So what our lab is doing is we're developing some digital means of doing this using sequencing or chip data. So I'm doing this using gene expression. Other members of the lab are doing it using methylation, but I'll be talking about gene expression. So the basic idea is that if you have some sample, a heterogeneous sample of unknown composition, and you also have the expression profiles of purified cell types. So if the heterogeneous sample is a combination of these cell types, you should be able to explain it as a linear combination. You should be able to do a linear regression and get a decent answer. And so fairly simple. So let's test it out. We have this beautiful mixture that Dennis Montoya made up. It's a combination of human immune cells. And so we have a nice array of some samples that are mostly T cells, some samples that are mostly monocytes. And we also have several reference data sets that we've collected from various sources. This includes ENCODE, Blueprint, and some other ones. And so what we do here is we kind of apply the simplest version of this idea of doing linear regression to estimate the cell type proportions. We start by normalizing. That is a necessary step. You kind of have to normalize each column of your data set. So each sample of unknown proportions and each purified cell type, you want those to have roughly the same distribution for the linear regression to work. And so if we apply this, we get an answer that looks like this. So these are estimated proportions on the right. These are actual proportions on the left. And I consider this to be pretty bad. We're at about 16% error. So on average, our estimates are off by about 16%. We completely miss these cells, and we seem to be unable to distinguish between CD4 T cells and CD8 T cells. And so what I propose is we do some things to improve this pipeline. This is kind of the simplest implementation. I say that we have these two steps, which I will talk about now. The steps are signature gene selection and row scaling. So signature gene selection, the idea here is fairly intuitive. Most genes in your data sets that you're looking at aren't going to be particularly useful for deconvolution. So, you know, a lot of housekeeping genes are such, their expressions are pretty flat across different cell types, and it can be much more accurate to tease out genes that are highly expressed in one cell type and lowly expressed in others. So a good example of this might be a keratin gene. It might be more highly expressed in keratinocytes than any other cell by a factor of 100 times or more. So if we pick out genes like this and use only those instead of the entire reference, we can improve accuracy and also improve speed, actually. That's speed hasn't really been an issue we've been running into, but it is an effect. We can get, this makes it faster. And there are many ways that we can do this. We've tested out a variety of them. We're currently using one of two. The first one is maximum z-score. So the idea here is you look at the tissue, the cell type in which your gene is most highly expressed, and you kind of look at that as one value, and you have the distribution of the rest, and how far away that value is from the distribution influences whether or not you choose it as a signature gene. The other method is a bit more nuanced. It has to do with this idea of information entropy, which comes up in a lot of fields like data compression. In some fields, they call it participation score. But the idea is similar. You're picking out genes that are highly expressed in one tissue and tend to be lowly expressed in the others. And another parameter that you can tune is how many signature genes you choose. We've generally been choosing around 50 for each cell type. It varies though. So let's say that we have a number of reference data sets, Blueprint, the Human Primary Cell Atlas, and Code. What I did was I made a large number of synthetic mixtures using one reference. In this case, it was Blueprint. And then I tried to deconvolute it using the Human Primary Cell Atlas and see if I can reclaim the proportions I used. And looking at the accuracy of that for these parameters, we see that we find a pretty good solution if we use z-score on 100 signature genes. And so we apply this solution to what we did before on Dennis's mixture, and we see that we get an improved solution. It's still not perfect, but we still miss T cells, but we do have limited ability to detect CD4 T cells and CD8, and the error gets a little bit better. It was about 16 before, now it's about 13 and a half. So that's the first thing we can do to improve our predictions. The second thing is this thing row scaling. And the idea here is that even after normalization and signature gene selection, you can get genes that look like this. So these are two genes that were selected as signature genes. The first one for monocytes, the second one for natural killer cells. And ideally, our system would use both of these. They're both good signature genes. But one thing that you'll notice is that the top one is kind of much more highly expressed everywhere. And so if we put this into the linear regression as is, it's going to kind of see it more like this, where it puts a lot of weight on this first one, and the second one is more or less ignored in comparison. So the goal of row scaling is to transform these vectors of expression values such that this effect is lessened. And so there's a few ways that we can do this. Kind of the simplest way is we just take the sum of each row and divide each value by the sum. The effect of this is that each row sums to 1. And so all rows are kind of treated equally. So that would kind of be an extreme way of doing it. But maybe that's too much. Maybe it's better to go kind of halfway and divide it by the log or the square root. So this serves to kind of rein in the higher values, but not by as much. Also, a slightly more complicated one is this linear transformation method, where essentially we take the vector of values, we set the minimum one to 0, and set the maximum one to 1, and then the other values we scale linearly between. And we can do these transformations either on the reference and the mixture separately or jointly. And what I mean by that is we can kind of perform the same transformation on the samples and on the cell types, on the purified cell types, or we can kind of smudge them together, do one transformation, then tease them apart again. So there's multiple ways that we can do this. Similar to with signature gene selection, I created a bunch of synthetic mixtures and tested on all of them, and tested all these methods. And the patterns are not as clear. One interesting way that the conditions can vary here is the number of samples you get. So when you're using the diversity among the samples to determine how you scale them, it's very different if you see 5 samples versus 100 or 1,000. And so the optimal method does vary depending on these conditions, but one solution that always seems to be good, if not the best, it is at least close, is this linear transform method jointly. So we apply this solution, and if we do it on the mixture we made up in the lab, we get this answer, which is looking much better. We have, our error is much down, and we detect T cells correctly, and we're able to distinguish between CD4 and CD8 T cells. And so that's pretty much the state of our program. future directions, some things that, some information that we could be utilizing to improve our predictions. We're not currently really utilizing replicates among the same cell type, and so I think when we compile our references, filtering out genes that where their expression varies in the same cell type, I think that would probably improve our results. Another thing we can do is in the linear regression, there's the error term. In addition to, we're expressing our observed expression in the sample, in our heterogeneous sample, as a combination of purified cell types, and then also an error term. Our model is currently somewhat blind to how big that error term is, and I think that's something we can also utilize. Lastly, a lot of what I've been developing here can theoretically be applied to identify clusters of cells from single cell data, which is something that a lot of people are working with and I think could be useful. That's pretty much it. My acknowledgements, thank you, Matteo, for being very helpful in your guidance. Dennis has also been very helpful, especially with generating these mixtures. And David Lopez has also been a member of the lab that has helped me out a lot.